Hello everyone, this is Dr. Madhuri Deshmukh. Biocell Tutorials welcomes you. Today we are going to start the lecture on structure of anther. So let us start. Structure of anther, or it is also called as a microsporangium, as the size of the spores are very small, so it is called as a microsporangium. It is bilobed and diathysious. All I will show you here diagram. Just wait. This is the anther. Anther is connected to the filament and collectively it is called as a stamen. And we can observe here, this is the dioecious structure, diathesis structure. It's showing the two lobes, and these two lobes, when we take the transverse section of this anther, we can observe here four pollen sacs. Okay, so let us start. A longitudinal groove separate the theca in a cross-sectional anther is a tetragonal structure consisting of the four microsporangia in two lobes. Later, two microsporangia of each lobes fuse as a pollen sac. Structure of anther. A microsporangium is a circular and surrounded by four layers. Outer layer is epidermis, which is followed by endothecium, then middle layer, and the innermost layer is tapetum. Outermost layer, just wait. Outermost layers protect the pollen and helps in dehiscence of anther to release the pollens. Anther wall. Epidermis is the outermost layer of anther, which is single cell in thickness. The cells of thin layer divide only anticlinically in order to cope up with the growing anther. This is the structure of flower. Here, stigma, style, ovary, and ovary having ovules. This is collectively called as a female gamete or pistil. Yellowish colored anther, which is connected to the filament, and this is collectively called as a stamen. Red colored petal, then green color sepals, then this one broad portion is called as receptacle and pedicel. Okay, now let us discuss the middle layers. Next to the endothecium, 1 to 3 layers of the middle layers, all of them degenerate at the time of meiosis in microspore mother cell. Tapetum is the innermost layer of anther wall. It is composed of a single layered cells characterized by dense protoplasm and prominent nuclei. Depending upon the behavior of tapetum is of two types, that is the amoeboid tapetum. It is of the primitive type later during the drying up process of anther. Periplasmodium hydrates and deposit as the trephine on the wall of the pollen grains. Then the another one is the secretory tapetum. Secretory tapetum cells remain attached to the middle layer till the development of the pollen grain. It is more common among angiospermic plants. Now let us discuss the function of tapetums, which are the major function of this tapetum. It provides the nourishment to the developing pollen grains. Also, it helps in formation of exine. Exine is the outermost layer of the microspore or pollen grain. Now, sporogenous tissue. Primary sporogenous tissue give rise to the microspore mother cells. Some of sporogenous cells remain non-functional and serves as a food material for developing microspore. Microspore mother cell undergoes meiosis to form the microspore tetrad, which separate out to form the microspore or pollen grains. This process of formation of microspore from microspore mother cell is called as a microsporogenesis. So now see here microsporogenesis. The what is microsporogenesis? The process of formation of microspores from the pollen mother cell through the meiotic cell division is nothing but the microsporogenesis, as already we have discussed here. The cells of the sporogenous tissues undergoes meiosis and forms the microspore tetrad arranged in a cluster of 4 cells. As each cell of sporogenous tissue has a potential to form the tetrad, so each cell is a microspore mother cell. On maturation and dehydration of anther, the spores dissociate and develops into the pollen grains. Pollen grain is nothing but the male gametophyte. These pollen grains are spherical in shape. They are two layered, outer layer and inner layer. The outer layer is called as a exine and the inner layer is called as a intine. This exine is consist of it is made up of sporopollenin and which is resistant to the organic matter, which withstand to the high temperature, acids and alkali and enzymes. It has also the prominent aperture. Aperture means hole. That is called as a germ pores, where the sporopollenin is absent. Intine, another one layer or wall of the pollen grain is intine. It is thin, continuous layer made up of cellulose and pectin. Sporopollenin is absent in the inner layer intine. Pollen grains, male gametophyte. Just wait. Male gametophyte. Pollen grains, cytoplasm is surrounded by plasma membrane. Mature pollen grains has 2 cells, that is the vegetative cell and generative cell. Vegetative cell is bigger in size, abundant food reserve food material, large irregular nucleus, and generative cell is small, comparatively small. It is spindle shape showing dense cytoplasm and nucleus which floats in vegetative cell cytoplasm. In 60% of the species, pollen grains are shed in 2 cell stage, whereas 40% species shed in 3 cell stage in each generative cell. And generative cells divide mitotically into 2 male gametes. Now see here transfer section of this TS of anther showing outermost layer epidermis. This one endothecium. Endothecium is followed by 2 to 3 layered structure, middle layer, and the innermost layer is the tapetum. Tapetum covered with the pollen grains. And here if you take the transfer section of this anther, this bilobed anther showing the tetrad structure, 4 sacs are showing on each corner. Okay, thank you. Hope that you have understand this all these about the structure of anther. If you have any queries or your doubts regarding this, you can comment me in comment box. Thank you. Have a good day.